I request all participants to mute on their video and microphone, please. And we can start now. Thank you.
Good morning and good evening, everyone. Welcome to today's webinar on Synthea Database Overview and Bioresearch Monitoring Clinical Data Reporting. My name is Sampada and I will be your moderator for today's session. We have a great lineup of speakers and the topics planned for you. So let's get started immediately. We will have a time for the questions at the end, so feel free to jot them down as they come to your mind. Now, I would like to introduce our first speaker, Suraj and Alekhya. So Suraj and Alekhya, please introduce yourself and then you can start your presentation.
Okay, Sanpada, thank you. Hi guys, my name is Suraj Pujari and I'm working as a statistical programmer at Synthea Data Sciences.
Hi everyone, good morning and good evening. I'm Alexia Itlam Setti. I'm a statistical programmer at Synthea Data Sciences Private Limited. Yeah, Suraj, please start.
So, hi everyone, thank you for joining today. My name is Suraj and my colleague Alekhya. We are going to present or talk to you about Synthea, a synthetic patient data generator. So let's get started. The agendas for today's presentation are: first, we will see the introduction of Synthea. Then we will see about the features of Synthea. What are the applications of Synthea? What are the technologies and systems required for the Synthea to generate the new data? What are the inputs required for the Synthea? And how to get started and how to get involved in the Synthea. And in the end, we will see about the licenses. All this information in this presentation is available in the link which is provided here. So you can also check the link, that is Synthea GitHub page. Next slide, please. Now we will see the introduction of the Synthea. What is Synthea? Synthea is an open source synthetic patient data generator, and this Synthea tool is used for the purpose of modeling and analytics. This tool is very useful for the researchers, educators, and developers who need realistic healthcare data without the privacy concerns associated with the real patient data. The main objective of this presentation is to provide the information about Synthea, its features, the system required for the Synthea to generate the new data, and how we can generate the sample data with the help of Synthea. Next slide, please. Now we will see about the features of the Synthea. There are total four features of Synthea. The first feature of Synthea is, with the help of Synthea, we can generate the fully synthetic set of electronic health records of the patient's data with more precision, realism, and scalability. With the help of Synthea data, we can also ensure the data integrity and accuracy. Synthea can generate large volume of synthetic data very efficiently, which is used for the robust testing and research purposes. The main important feature of the Synthea is the Synthea data is anonymized to maintain the privacy of the PII, that is personally identifiable information. The last feature of the Synthea is generated Synthea data can be available in different formats such as text formats, CSV formats, other healthcare formats such as HL7 and fast healthcare interoperability resources. Next slide. Now we will see what are the key applications where Synthea data can be used. Synthea data can be used in academic research. In academic research, researchers can explore the validity of various studies, and it also encourages further investigation in population health. In healthcare systems, Synthea data can be used to assess the performance of healthcare system, and it is also used to evaluate the effectiveness of new processes and technologies. In machine learning and artificial intelligence, also Synthea data can be useful. This Synthea data can be used to train and test machine learning algorithms and artificial intelligence systems in the healthcare domains. With the help of this Synthea data, we can predict the disease diagnosis, treatment recommendation, and patient risk assessments. Next slide, please. In medical education and training purposes also, Synthia data can be used. This data can be used to create realistic case studies and training scenarios for the medical students, and it is also useful for the other healthcare professionals. This uh, this helps them to improve their diagnostic and treatment skills. In healthcare policy analysis, epidemiological studies, and clinical research, also Synthia data can be useful. This allows the researchers to explore the various scenarios and test hypotheses without compromising the privacy of the patients. Next slide, please. Now we will see what are the technologies Synthia can uses. Synthia is an open source application and it requires Java JDK, that is Java Development Kit. The version of this Java Development Kit should be 11 or newer, which is used to generate the new data. We also need Excel, which is used for the used to access the downloaded data in the CSV format. Next slide, please. Now we will see what are the system requirements to generate the Synthia data. Synthia data can be generated with the help of Java Development Kit, that is Java JDK. The version of this kit should be 11 or newer. It is always recommended to use a long-term support release of Java. For example, version 11 or version 17 should be preferred. Now we will see the inputs required for the Synthia to generate the data. Synthia data can be generated with the help of Synthia dependencies jar file. This jar file can be downloaded with this link which we have provided, and we can generate the Synthia data. There is also another way where we can download the sample data. This sample data is available on the Synthia website. The link is provided at the end of this presentation. Next slide, please. This is the hub part of this Synthia presentation. So till here, if you have any questions, then please put it in the chat. Okay, meantime, if you don't have any questions, so we will continue with the another half part of this presentation. So my colleague Alekhya is going to present you about the half part of this presentation. So Alekhya, it's over to you. Thank you.
Thank you, Suraj. Yeah, once again, good morning and good evening, everyone. Today I would like to talk about how to use Synthia, like how to install the Synthia, how to generate the data using Synthia, and how to process with the configuration parameters, and we can see how to process the data after how can we process the generated data as well. And Before starting the installation process, let me walk through briefly what is Synthia. Synthia stands for Synthetic Health. It is an open source project which simulates patient from birth till death. Like it produces high quality, realistic data, but not like real data. In Synthia, we are having around more than 30 modules, 540 different clinical concepts as well. And Synthia records we can use for any sort of non-clinical use, secondary use, like without no restrictions. It has no restrictions. It is has no legal, no cost, and also has no privacy security restrictions as well. And also Synthia, it is not hampered by HIPAA too. And we'll see like why Synthia. See, we are having high demand on EHR data sets, electronic health record data sets. Like it is very difficult to access due to lack of access. And see, even though they are like anonymized, records are being bought and sold, and also the real data records are like they carry some privacy, confidentiality, consent, and some legal risks. So like for example, see like for clinical testing or for interoperability for non-clinical secondary users. Simply like realistic data could be sufficient, so but not like real data. So if you want really a good realistic data, you can go with Synthia. Now let me start with the installation process. Uh, we can download the Synthia in using a GitHub page. In GitHub page, we can see the Synthia link to download, and clearly they'll mention some of the instructions how to download the Synthia. And simply following the instructions, try to download the Synthia. And then, as Suraj mentioned, we need some dependencies like Synthia dependencies.jar, and we need Java JDK. The newer version or 11 version is uh, it would be sufficient. And then we need some git version control, and then we'll see like how to generate the data using Synthia toolkit. Here, uh, next slide please. Yeah, this is the Synthia toolkit which used to generate the synthetic data. Here we have provided the link for Synthia toolkit as well. You can access from that. And see here we are having two options like patient viewer and the Synthia customizer. In patient viewer, like you can go and search with the JSON files. You can go and search with the content of the JSON files. In Synthia customizer, further we are having guided mode and advanced mode. I suggest like if you're like first time users, you can go with the guided mode. It has some basic configuration settings with some series of questions. And in advanced mode, if you are like aware of what customization options you need to give to generate the data, you can go with the advanced mode. Yeah, next slide, please. See, here in guided mode, like we have some configuration options with a series of questions, and we can after here in this presentation, I'll explain how to generate the data using guided mode. So, if you select the guided mode, after that, it will show some of the formats which are available in Synthia, like HL7 file format, CSV format, fire bulk data, and JSON file, HL7 fire STU3, HL7 fire DSTU2 format, text format, and CPCDS, CMS, BFT, and CDW formats. You just select whatever format you need to get the generated data. Yeah, next slide. If you go and select the data format you need, it will show some of the data requirements you need to get access. Like if you want to generate the data with a certain demographics, you can go with that particular option. Also, you can generate the data with some certain clinical conditions. Like you can go with the conditions part, you can go with the medications. And if you want to generate data with a certain observations, with a certain procedures, you can go with that particular option. Also, you can generate data with a certain geographic location as well. You can recreate the same exact population multiple times also. So after selecting the data requirement you needed, it will show like some basic settings as you see in the figure. See here, I'll just explain with the demographics. If I give the population with the specific demographics option, it will show some basic settings and some demographic settings. In basic settings, it is having population section. In population, you can give the number like if you want to generate data with 100 patients, you can give the number 100. If you want to generate data with 1,000, 10,000, it depends on you. How many people, how many patients you need, you can give that number. And in demographic settings, you'll see some basic configuration settings like gender, age minimum, and age maximum. So if you want to generate data with men, you can give M option M. If you want to generate data with women, then you can give female F. Okay, and if you want to generate data with the children like 7 to 20, 7 to 12, like that, you can give their range with a minimum 7 and maximum range 12. So after giving all these demographic settings, we'll see the next set of options. Next slide. Yeah, here see after giving all the basic settings, all the demographic settings, all the prerequisites required for this, we'll see the setup to run the Synthia. Basically, we have three different setups like Docker setup, basic setup, and developer setup. Here, I'll explain how to use the basic setup. Also, we can go with the Docker setup, which has like differences with the prerequisites needed between Docker and basic setup. Like for basic setup, we can have Java JDK, Synthia dependencies.jar. Whereas in Docker, we need, like for example, if you're like a Windows or Mac users, you need to install Docker Desktop. And then if you're like Linux user, you need to install Docker Engine. And it is also having some difficulty with configuration options. So for now, like first time users, you can go with the basic setup. Like if you run Synthia with basic setup, in that page itself, it shows some command line based on your configuration parameters you have given. Okay, you can simply copy that command line and you can paste over in your command window or prompt window. Next slide, please. See, you can open your command line prompt or terminal window and run the following command you have uh, got in that particular page, Synthia toolkit page. And then, for example, see here I have given Java jar Synthia with dependencies dot jar and population with 100. I required only 100 patients, then I'll give 100. If I required only male, then I'll give G with M. G stands for gender over there. And I want only 25 to 49 population. Then age with the minimum 25 and maximum 49 range is given. And if I want to generate data in CSV format, I can use option exporter.csv.export equal to true. And if I don't want like in a CSV format, if I want in other formats like fire format, I can use the true option with that exporter, like exporter.fire.export equal to true. If you don't want to generate data in fire format, you can use simply false option. So once you run the Synthia in your command line terminal windows, it will process the downloading process. It shows some modules, and after like downloading everything, you'll have a creating a output folder in your system. Like it inside, it is also having another output folder with the format you have given. Like if you want a CSV format, it will show some CSV format related, and all the domains will be mentioned in that output folder. Like synthetic data will give conditions, medications, patients related information, and if patients is having allergies, allergies related information will be presented, and care supplies like that. See, for example, patient is having like MRI, then it will go with the imaging study. It will be in that imaging study domain like that. So you can get access to your contents in the output folder, and at the same time, you can go with the patient viewer option also. There you need to select JSON files. So if you select your JSON file, you can simply view your contents of that particular JSON file. So it contains each and every synthetic patient details like their conditions, their medications taken, what are the procedures they have used, who are the providers of that particular patient, and what are the care plans they have taken. All these things. Yeah, this is about how to generate the data using Synthia toolkit. Now we'll see how to process the data after generating. Next slide, please. Yeah. Um, like we pre-process the data, like such as cleaning, transforming, and merging the data sets to ensure data quality and compatibility within your analysis environment. Usually, if we generate the data, we may get some missing values, and like which may lead to bias in our analysis system. So we need to deal with these missing values. We have some imputation methods to deal with the missing values, like mean median imputation method, mode imputation method, linear regression imputation method, k nearest neighbor imputation methods, and uh, mice technique, mice imputation method as well. So we'll see what is like mean or median imputation method. This method, like it replaces missing values with the mean or median of that particular observed values within the same column. It is a very simple and effective. And for example, see if you are having data with ages like 25, 30, 40, 35, and one missing record, and how to deal with that missing value, like how to replace that missing value. I can simply do the mean of that observed values within the same column or median of that, and you can replace with that mean value. Next slide, please. Yeah, mode imputation. See here in mode imputation, it is uh, for categorical data, mode imputation replaces missing values with the most frequent category in the column. For example, if you are having red, blue, green, and some other colors and some missing values, here it checks with the most frequently available color and it will replace us with that. Here in our data, if you are having more red color, like that missing value will just replace with that red. So now, K nearest neighbor imputation. It is like machine learning algorithm technique we use to deal with the missing values. It is based on the values of its nearest neighbors. It calculates distances between data points and fills in missing values using that average of nearest neighbors values. For example, if you are having person's age is missing, if you are having variables with gender, education, income, and age, and person's age is missing, then we can use like K nearest neighbor imputation method. It will take average age of those similar individuals using that as the imputed age for the missing value. It will base on that other variables like income, education, and gender. Based on those variables, it will just impute the age missing value. Next slide, please. Linear regression imputation method. It is very effective, like it applies for linear relationship but not capture non-linear relationships. And if missing values are estimated using a linear regression model based on other variables, like if you are having data set like income, education level, and age, and in this income is missing, it just used a linear regression, like it assumes that whether income is related to education and age, and it will build some regression model using education and age variables as predictors, and it imputes the missing value based on that prediction method. Next slide, please. Yeah, multiple imputation by chained equations. It is a mice technique. It works on iterative model. It is an iterative imputation method that improves missing values by modeling each variable with missing data as a function of other variables. Like it works iteratively cycles through variables and imputes missing values based on conditional distributions. For example, if you are having variables like height, weight, and age, and in this weight is missing, here how can we use like mice technique? First, it imputes weight based on height and age variables, and then it imputes height based on weight and age variables, and finally it imputes the age based on height and weight variables. So it iteratively cycles the variables and imputes the missing values. Yes, these are the techniques we use. Some of the techniques we can use to deal with the missing values. Next slide, please. Analysis and research here. Synthia can be used to for various analysis, various research studies, or various for simulations also. It can apply for statistical methods, machine learning techniques, and also extract insights or test hypotheses as well. Next slide, please. Yeah, we can download Synthia using this zip file in Synthia website in Synthia website in GitHub. You can extract that file and you'll see various domains like patients, conditions, observations, medications, procedures, and care plans, and allergies related information as well. Next slide, please. See, here in this section, we have mentioned all the links related to GitHub, related to modules, related to Synthia data, how to download the Synthia data. And we have given the Synthia Maitri page as well. You can go and access over that, and you can get some more additional information from these GitHub pages. Next slide, please. Synthia, it was copyrighted with the Maitri Corporation. It was licensed with Apache license. Yeah, by this, I can say that we have given like how to install the Synthia, how to generate the data, and how to process with the generated data. Yeah, next slide, please. So finally, if you want to get like some more information, if you want to get involved in efforts of Synthia, if you are having any questions with patient data generation process, you please contact with contact@sumtersds.com. Yeah, thank you.
Thank you so much, Suraj and Alekhya, for your insightful presentation on Synthia Database Overview. Now, I request audience, if you have any question, you could ask it. So I can see there is a one question. So does the data follow CDISC standards? This is a question from the audience.
Uh, Actually, it follows FHIR standards. We can check interoperability between FHIR and CDIC standards as well using the Synthia data, but it follows FHIR standards.
Thank you, Alekhya.
Yeah.
Any other questions on this topic? Audience, I request you to please post your questions in the chat box. Yes, so there is a one more question. Okay, so it's a synthetic data. How much can we rely on it and how to avoid the biased data? So this is a question from the audience.
Yeah. As it is synthetic data also, it is have we can use without any restrictions. Uh, we can get access as it is an open source. We can see in this it is given like it is giving the patient's information from the birth till that it is having all the progression and disease related information in that. And yeah, we can use for research and analysis purposes as well.
Yes, there is a one more question. So can you have a synthetic include abnormal lapse reading in the output?
I guess yes, it can include some abnormal values as well. Like it is like for researching purpose, like we can use.
Thank you, Alekhya. Thank you, Parag. Any other question from the audience? Okay. Now we will hear from our next speaker, Kala. She will be representing on the topic Bioresearch Monitoring, Clinical Data Reporting. So Kala, please introduce yourself and after that you can start your presentation.
Thank you, Sampada. And thanks to Alekia and Suraj as well. That's a lot of information. Well, to introduce about myself, I am Kala Vonda and I'm a statistical programmer at Sumter's Data Sciences. Today, the second topic of this webinar is going to be the BMO clinical data reporting requirements. Can we move on to the next slide? Yeah. So BMO stands for Bioresearch Monitoring. Throughout, we'll walk through these coming slides. and go through each one of them, to see what BMO is all about, what are the objectives of BMO, and what is the scope of BMO, and also the clinical data reporting requirements for BMO. And also we'll go through a few of the case studies as well, presenting the different options for the BMO listings that can have. Next slide, please. So BMO stands for the Bioresearch Monitoring Program. The introduction of this BMO program by FDA was initiated when FDA conducted a survey of the clinical study trial conduct. Out of the several outcomes from the survey, major deficiencies were towards the subjects' welfare, safety, and overall data integrity, which were under severe impact due to a huge bias. Well, the potential source of this bias in clinical studies is majorly due to a financial interest of the clinical investigator in the outcome of the study. It could be due to several reasons. Depends on the way payment is arranged. It could be proprietary interest, it could be equity interest. Whatever the mode of payment is, there was a huge bias due to the financial interest. So these, along with several other deficiencies, established a need for the FDA to be given the right to audit these clinical trial research sites. So they came up with this BMO program. It's an agency-wide program of on-site inspections and data audits that were designed to monitor all aspects of the conduct and reporting of the FDA-regulated research. The main scope of this BMO program is to protect the rights, safety, and welfare of the subjects, to verify the accuracy, reliability, and data integrity of be it clinical and non-clinical trial data that was submitted to the FDA, and to assess the compliance with FDA's regulations governing the conduct of the trials, be it including the regulations for informed consent and the ethical reviews. Next slide, please. So let's talk about what products fall under the purview of the BMO audit. Any drugs, biologics, medical devices, food and tobacco products, and veterinary products, any products that needs an FDA approval needs to go through this program. And which companies are subjected to BMO audits? Both domestic and international companies, and any entities that are involved in an overall production of a product that needs an FDA license has to go through this BMO audits. Be it non-clinical testing laboratories, they are tested for good lab practices. Clinical investigators are tested for good clinical practices. Sponsors, CROs, clinical trial monitors, institutional review boards, and bioequivalence facilities. All of these companies will be subjected to the BMO audits. Next slide, please. And when they initiated this BMO program, FDA started collecting a lot of information from several sites of different studies. That was tons of information for them to go through. So eventually, they started calling on sponsors to provide them the review aid, which made them to streamline the entire process. So they came up with a technical conformance guide, which provides a consolidated, organized set of data that was drawn from the entire database of that study and put together in 3 parts so that it's easier for them to streamline the entire auditing process to check for the reliability. So CDER will use this information along with the NDA application or BL application, whatever application they're applying for, the BMO data will be reviewed along with that. This is organized in 3 parts. Part 1 includes clinical study level information. Part 2 includes subject level data line listings by site. And part 3 includes the summary level clinical site datasets. Next slide, please. So moving on, we'll go in brief about what these parts and what the data will be put into these parts. Part 1 includes the clinical study level information. It's a comprehensive and readily located list of clinical sites. as you see in this presentation, we are seeing a picture of the table that has all the information of the sites, site ID, name of the clinical investigator, and the address and the contact details of the clinical investigator site. And information should also be provided if there are any changes towards the site, if the site is terminated or if there's a change in the clinical investigator. All this information should be provided in this table. If at all, if there are multiple studies that are pivotal, then we need to include another table for this. Along with this, we should also provide another list of, can we move on to the next slide? Yes, thank you. So along with the earlier listing, we should also include a table which lists all the documents that are available for that study, including the monitoring plans, training records, data analysis plans, or all the other documents that should be included in a table, and also the location of these documents, whether they are in TMF or if they're at a particular location, the site where it was and that address all should be provided in this table so that it's easier for CEDAR to plan their inspections and reach out to that particular site and faster the whole inspection process. And along with that, we should also include the protocols, protocol amendments, and associated versions of CRFs, and the final versions of annotator CRFs. If at all we are including the BMOS data reviewers guide, that should also be included, and a note should also be put into the CSR's appendix. Can you go to the next slide, please? So that concludes part one. Now moving on to the part two. Part two includes all the subject level data line listings. Now the data that was collected from the CSR was organized into these 10 listings by site. We will also discuss a bit brief about these individual listings in detail in the coming slides. Let's go. So the first listing is the consented subjects. This listing provides the information by subject, by clinical site listing, whichever subjects were consented to enroll in that study. If at all, if there are screen failures that signed informed consent and were not included, they also should be included. If at all, if few subjects consented to the study but were not randomized, or if they're randomized and are not given treatment, the specific reason should be mentioned in this listing. So the overall information regarding the consented subject should be provided in this listings by site. And the second listing is the treatment assignments. This should include all the treatment assignments to which the subjects was randomized to per that site. If the subject receives a different treatment, the actual treatment that the subject has received to should be mentioned in these listings. And the third listing includes all the discontinuations informations of the subjects that have discontinued during the run-in period or from the study treatment or from the study completely. All this information, whoever have discontinued from the particular site should be included in these listings. Next slide, please. So the fourth listing is about study population. This identifies the protocol-defined study population in which each subject was subjected to analyzed to. It could be dependent upon the protocol. It could be a intent to treat analysis population. It could be per protocol analysis population. Whatever analysis populations that the subject has been through the entire study should be included in this listings. For subjects that did not meet this inclusion criteria, the reason should be provided for that. And the fifth listing includes the inclusion and exclusion criteria listings. This includes whether each subject met the inclusion and exclusion criteria that was defined as per the protocol. And the sixth listing is the adverse events. It includes all the adverse events related information, be it non-serious adverse events, serious adverse events, deaths, when this event has occurred, what's the severity, whether it was considered serious by the clinical investigator or by the sponsor, and what is the action taken. Did it lead to the discontinuation of the treatment? All the information related to the adverse events should be mentioned in this listing by subject by site. And the seventh listing is about protocol deviations. This includes all the protocol deviations, description of it, whether it is major or minor, or the sense important or unimportant, that are decided by the sponsor. Next slide, please. The eighth listing is the efficacy endpoints. It contains the primary and key secondary efficacy parameters or events. If at all the study's efficacy parameters or endpoints are based on derived or calculated endpoints, the raw data that was used to derive or calculate those endpoints should be provided. Be it lab results or imaging that was used, those should be provided here. And if at all, if the efficacy related endpoints is a clinical outcome or an event, then the events should be mentioned, and the date of the event, and adjudication date, and the adjudication outcome result. All this information should be provided in these listings. And the ninth one is the concomitant medications. Medications as specified by the protocol, a date started, stopped, dose and route of administration, and reason of administration. All that information should be included in this listing. And the 10th listing is about safety monitoring. It contains the results of tests, lab results, ECG results, PC data performed for safety monitoring based on the protocol. If the study endpoints are collected as clinical events, similar to the efficacy endpoints, listings should be provided that includes the clinical event, date, and when adjudicated, and the adjudication outcome. So this sums up the listings that were provided in the part 2. In the end of the presentation, we will go through case studies where there were different formats of presenting the same set of listings. So moving on for part 3, we have the summary level clinical site dataset. This is a single summary level clinical site dataset. This is named as ClinSite. That contains the data from all the pivotal studies used to support safety and efficacy in the application. This summary is based entirely on the safety and efficacy related population. So we will discuss in detail about the various variables that were used in this data set. So to start with the site-specific data, the variables that were included in this data set are sponsor count, what is the name of the sponsor and the number of sponsor counts, and the IND under IND, NDA application number, BLA, subname, if the application is a BLA or subname, supplemental. And then we'll have the study ID and title variables as well. Moving on to the site-specific information, the entire details about the site is listed here. First name, last name, initials, name of the investigators, phone, fax, and all the contact details and the address of the clinical site is mentioned, is put together in these variables. Now, financial disclosure variable is a very important variable. This specifies the financial amount that was given to the clinical investigator. Title 21, Part CFR Part 54 requires an applicant to disclose certain financial arrangements between sponsor and clinical investigators. Now, this information, in conjunction with information about the design and purpose of the study, as well as the information that was obtained through on-site inspections, all this information together will be used by the FDA in the agency's assessment of the entire reliability of the data, whether and have a conclusion regarding the possibilities of bias from the clinical investigator or sites. Moving on, we'll have site ID, ARM, and cohort variables where we can add the values of those site ID and ARM and cohort in these variables. And screen variable will have the number of screen subjects per that site per treatment arm. And serious adverse events, non-serious adverse events, death, number of deaths happen per site per treatment arm. All of these counts is located in safety variables. Important deviations and non-important deviations, discontinuation from the study, discontinuation from the treatment, all these are the counts of subjects by site and by treatment arm. Can we go to the next slide, please? Thank you. And moving on, we have another set of variables for this clean site data set, which are site-specific efficacy results. We have the SAF POP, EFF POP, both are the number of subjects in safety population and efficacy related population, respectively, by site, by treatment arm. And we have TRT EFFR1 and TRT EFFR2. Both are the summary statistics for each primary efficacy endpoints by treatment arm, by site, by a safety population and efficacy population, respectively. And then we have the endpoint variable. Endpoint variable usually describes the primary endpoint. It's a plain text label. It describes what efficacy endpoint we're talking about, and what TRT-FFR1, TRT-FFR2 is pointing to. An endpoint type variable comes handy when we have multiple endpoints for that study. Whether it's a discrete endpoint or continuous endpoint, or some cases time to event endpoints, that can be mentioned here. For each endpoint type, there will be an extra additional record by site, by treatment arm, by endpoint and endpoint type. And we do have the arm and cohort related variables. And sensor 1, sensor 2 variables are only applicable if we have a time to event endpoint. These will be included in the dataset, but they will be left blank or missing if our efficacy endpoint is not time to event endpoint. Can we go to the next slide, please? Moving on, we'll have an example how a clean site dataset will look like. This example shows how when we have one endpoint, we can see the study ID, title, sponsor count, all these variables provides information about the sponsor and sponsor related variables. It also provides the IND number and NDA application number. And we do have the site ID and ARM variables. If you can see, the SAF POP and EFF POP variables present the number of subjects that belong to the safety population and efficacy population. Number of screen subjects, number of discontinued from the study subjects, discontinued from the study, discontinued from the treatment, per that site ID per arm is given here. Moving on, we have the endpoint and endpoint type TRT-FFR1 and TRT-FFR2, sensor 1 and sensor 2. All these are efficacy related, and all these are efficacy related variables. And we do have the serious adverse events variables and non-serious adverse events, death, important deviations, non-important deviations, all these related to the safety related variables are provided here. And financial disclosure amount that we discussed earlier is the amount that the clinical investigator gained out of conducting that study was included here. And the first name, last name, and the entire address, contact details of the clinical investigator and the site is included here in the coming variables. Can we go to the next slide, please? This table provides the clean site data set where the study has multiple primary endpoints. You can see here the endpoint type has both binary and continuous, so that the endpoint type comes in handy to mention which endpoint type we are talking about, and if at all, if we have multiple endpoints. The records, there will be a separate record for each endpoint type. Can we go to the next slide, please? So that sums up all the part 3s of the overall BMO clinical data package. From now on, we will go see a case study. In one of the NDA applications to a FDA, the clinical study level information and the subject level information listings by clinical site is submitted by site, and by listing. You can see the example here. The listings was provided by site and by all the 10 listings were added. You go into one each listing, all the subjects information were included in that listing. This option A can be chosen if at all if we have a CSR template in handy. And that CSR template provides all the information, whatever information that the technical conformance guide and FDA is seeking out. We can reuse the same template to generate this option A sort of listings. We will go on to the next case study. Thank you. Where the sponsor chooses to present the listings in a different order. Here, the sponsor chooses to present the listings by site, by subject, and by listings. Earlier, the sponsor chose to pick by site and by listing and then by subject. But here, the sponsor chooses to go by subject profile and then uh, by listings. So what you can either pick option A and option B. It entirely depends on what options you have at your hand. Here, the sponsor already has patient profiles in hand. So they are already generated and contains the required contents that were asked for. So this order is also acceptable by the FDA. So that sums up the. Can you go to the next slide, please? Yeah, thank you. So that sums up the BMO clinical data reporting requirements. These BMO listings development has been integrated as part of the whole programming processes. Typically, these listings come at the mid to end of the trial when a majority of the data is available. And development of these BMO listings should follow the same programming processes, including validation and documentation of specifications. Adherence to the FDA guidance while following these BMO processes is very important. Next slide, please. We do have all this information available in FDA.gov. They were providing the Bioresearch Monitoring Technical Conformance Guide, and they also had BMO's Data Reviewers Guide completion guidelines. And we also have the draft guidance. All this information can be fetched from the FDA's site. Next slide, please. Thank you all for your time. Please let me know if you have any questions.
Thank you so much, Kala, for your insightful presentation on bioresearch monitoring, clinical data reporting. Now, I request audience, if you have any questions, please type it in the chat box. Yes, Kala, there is a question. subject level DLs, are these listings similar to the ones submitted for interim analysis or DSMB?
These listings can follow the same template, but we need to make sure whether whatever information they are actually seeking out for each listing was included or not. They want to see whatever information they asked for. They wanted to see in these listings. We can follow the same template. We can follow whatever we have, but we need to make sure that these are organized in the way they asked for, and the information that they asked for were included in these listings.
Thank you, Kala.
Yeah.
The next question is the BMO are the site specific listings. Okay, so this is nothing. Okay. Some more explanation on this question. Thank you, Parag. The next question is what technologies or the methodologies are being utilized within the BMO project?
We can use any macros that we have already that we used for CSR generations. We can use the same macros and all.
Thank you, Kala. I request audience, if you have any questions on any of these two webinar topics, you can ask it here now. Okay, the next question. What measurable outcomes or the benefits has the BMO project achieved thus far?
Most of the times there wouldn't be any action indicator from the BMO's side. If at all, if they need some explanation or if at all, if they found some discrepancies, they would issue voluntary action indicated, or if at all, if they find some objectionable discrepancies throughout the conduct, they would issue an objectionable action indicated for. So what are the measurable outcomes and benefits? Benefits being to check for the safety of the subjects, data reliability, whether the data that was submitted was reliable enough, and integrate data integrity. Those are the benefits. Outcomes, it could be based on the conduct of the study and the data that was presented.
Okay, thank you so much, Kala.
Thank you.
Yeah, so if you have any more questions, you can reach out to us at contact@sumptuous-ds.com. A big thank you to our audience for their active participation and insightful questions. And of course, the special thanks to our speakers for sharing their expertise with us today. So remember, you can access the recording of this webinar on our YouTube channel. The link will be posted on our LinkedIn page. So stay tuned there for updates. Have a great day ahead. Thank you, everyone.
Thank you, everyone. Thank you for the
Thank
time.
you, everyone.
Thank you all.